In this topic, we're going to have a look at cancer. So by the end of this topic, you should be able to answer the questions: what is cancer? What are proto-oncogenes and tumour suppressor genes? What is a mutation? What is a carcinogen? What are the factors that increase the likelihood of cancer? What's the difference between benign and malignant? And what are the steps leading to the development of cancer? So the word cancer was first used by Hippocrates in 400 BC. He saw a similarity between the swollen veins radiating from a breast tumour and the legs of a crab. So the Latin for crab is cancer. Cancer is one of the most common diseases in developed countries, and it accounts for roughly 1 in 4 deaths. So what is cancer? It's a group of diseases caused by a growth disorder of cells. They are the result of uncontrolled mitosis. The genes that control mitosis and the cell cycle get damaged, so you have cancerous cells dividing repeatedly and out of control. This will result in a tumour, which is an irregular mass of cells. So cell division is controlled by genes, and most cells divide at a fairly constant rate, so that you have the dead or worn out cells being replaced. Now in normal cells, this rate is tightly controlled by 2 genes. You've got the proto-oncogene, which stimulates cell division, and then the tumour suppressor gene, which inhibits cell division. So what's the role of proto-oncogenes? These stimulate cell division. So in a normal cell, you have got growth factors, and these attach to a receptor protein on the cell surface membrane. And then via relay proteins and a nuclear protein, they switch on the genes necessary for DNA replication. So what's an oncogene? Well, when you have a gene mutation causing a proto-oncogene to mutate, it mutates into an oncogene. So here you can see that when a cancer-promoting agent, for example, UV light, causes a change in the gene, this is going to result in an oncogene, and this will cause cancerous cells to divide uncontrollably. So how do oncogenes affect cell division? Well, the receptor protein on the cell surface membrane can be permanently activated so that the cell division is switched on, even in the absence of growth factors. Or the oncogene may code for a growth factor that is then produced in excessive amounts, so you have excessive cell division being stimulated. Can you remember what the role of the tumour suppressor gene is? This has got the opposite effect to proto-oncogene, so it inhibits cell division. So what do you think is going to happen if you have a mutation in the tumour suppressor gene? So if the tumour suppressor gene becomes mutated, it's going to stop inhibiting cell division. So you have cell division increasing. And then you've got the cell cycle checkpoints. So these are different checkpoints that occur during the cell cycle, and they help to keep the different events in check. Cancer results when the G1, G2, and mitosis checkpoints fail, and thus cannot check if the DNA replication is correct and complete. So this means that the cell will continue to divide uncontrollably. So what's a mutation? A change in any gene is called a mutation, and mutations are not unusual events, but most of the cells that are mutated are either destroyed by the body's immune system or they have an early death. Now, cancerous cells, however, manage to escape both possible fates. This means that although there was a mutation in one cell, it's going to be passed on to the cell's descendants. So then what's a carcinogen? Now it's thought that a single mutation cannot be responsible for cancer, but you have several independent rare accidents must occur all in one cell. And a factor that brings about any mutation is called a mutagen. And if it causes cancer, it's called a carcinogen. Okay, let's have a look at the factors that increase the likelihood of cancer. You've got ionizing radiation, for example, X-rays, gamma rays, and UV light. These cause the formation of damaging ions inside the cells that can break DNA strands, and then ultraviolet light can actually damage the genes, and this is going to result in cancer cells. Chemicals. Many chemicals have been shown to be carcinogenic, and these damage the DNA. So here's a table of chemical carcinogens and the cancers they may cause. For example, diesel exhaust has been linked to lung cancer. Pesticides, lung cancer. Cigarette tar, lung cancer. Asbestos, lung and bladder cancer. Hair dyes, bladder cancer. Arsenic, lung and skin cancer. And formaldehyde, nose cancer. Some cancers in humans and animals are known to be caused by viruses. Now, the Burkitt's lymphoma is the most common cancer in children in Africa. So here you can see a child with Burkitt's lymphoma, and it's a cancer of the lymphatic system caused by the Epstein-Barr virus. There are other cancers that cause leukemia, and then you have the papilloma viruses which have been linked to cervical cancer. Cancer tends to be more common in some families than others, indicating a genetic link. In most cases, it's not the disease that is inherited, but the susceptibility to the factors that cause the disease. So there are some forms of cancers that do appear to be caused by the inheritance of a faulty gene. For example, an error on chromosome 13 can be inherited, and this causes something called retinoblastoma, which causes blindness. So can you remember the different factors that increase the likelihood of cancer? You've got ionizing radiation, chemicals, virus infection, and hereditary disposition. Now, you've heard of the terms benign and malignant. What do they mean? Well, you have a small group of tumor cells is called the primary growth, and it can have two types. You can have benign or malignant. Benign tumors do not spread from their site of origin, but they can compress and displace surrounding tissues. For example, warts, ovarian cysts, and some brain tumors. Now, malignant tumors are far more dangerous. This is because they can spread throughout the body. They grow rapidly and they tend to spread to other regions of the body. When they spread, this is called metastasis. Okay, let's have a look at the different steps leading to the development of cancer. So carcinogens cause a mutation in the gene that results in an oncogene. The oncogenes are thus switched on and the tumour suppressor genes are switched off. The cancerous cell does not respond to intracellular signals from other cells, so you don't have apoptosis occurring. Apoptosis means cell death. The cells continue to divide by mitosis. The immune system doesn't recognize the cells as foreign, so the cells are not removed. They continue to divide by mitosis. And you have a formation of a mass of under or unspecialized cells. The tumor eventually gets its own blood supply, and the cells can spread into the lymph. Then you have metastasis when the cells invade other tissues and you get secondary cancers forming. Okay, we've come across quite a few keywords which you need to learn. Oncogene, which is a gene that causes cancer. Mutagen, which is a factor that causes mutation. And then carcinogen is a factor that causes cancer. Apoptosis, which means cell death. Metastasis, where cancer cells invade other parts of the body. Malignant, cancer cells that can spread. And then benign, cancer cells that do not spread from their site of origin. And that concludes our lesson. The end.